Our last speaker of this session, this morning session, is Professor Terje Svingen from the National Food Institute and Technical University of Denmark, and he's one of the coordinators within the recently launched Encore cluster. That includes 5 EU-funded projects focusing on developing of new EDC tests and screening methods. And he will discuss one of the currently most debated topics around EDCs, namely new approach methods known as NAMs, and their potential and limitations in identifying and assessing EDCs. So the floor is yours.
Good morning. It's been some excellent talks. So I've been given the good and not so good task of explaining everything about new approach methodologies and how we're going to change the world in chemical testing. I need a couple of hours for that, but I have 10 minutes. To set the scene, what it's all about is, so the EU has this roadmap of phasing out chemicals testing using animals. They want to use new approach methodologies, which are mainly non-animal methods they are thinking. So how to do that? So I'm going to just put the scene and not tell you really how we're going to do it. I'm going to tell you a couple of challenges and then a couple of opportunities. In all challenges, there are opportunities. Now, I did not pick that yellow color. Something has happened. It was blue. So I was not drunk when I made this talk. But in any case, we'll see how it goes. And if I can work out which one to press. Here we go, blue. So this is just what everybody talk about. This is the life cycle of reproduction. This is the life cycle in general. My focus is a lot on the fetal stages, but all of it matters. And if you mess around with the fetal stages, you mess around with life. That's just how it is, because you bring it on with you. So here we go. Another thing I want to point out, and for some of you, this is like, yeah, I know that, but a lot of you might not. So when we look at the two sexes, male and female, a lot of us think that we are quite similar but quite different depending on where you look. So we have the same muscles, we have the same brain, we have the same this, we have the same that, and then we look down below in the reproductive system and it's totally different. That's fine, but you also have to remember that no matter what tissue you look at, we all started out exactly the same, with the same cells, the same tissues. So even the reproductive system looking quite different here, which you all agree. When we look back during development, they are actually not that different at all. So when you look at these adult genitals, the penis and the clitoris and the vagina and so forth, they are very different. When we start going back into development, tracing back in fetal life, they look more and more similar until you get to the starting point when we call it a genital tubercle. And they are actually identical. So this is quite important to remember, and I still frustratingly hear that, well, even experts say this sometimes, that we all start out as girls and then some become boys, which is frustratingly incorrect. We all start out as non-sex, and one becomes boy and one becomes girl, and that's because we're starting out all the same. So this is sort of setting the scene where I work on reproductive toxicology and how we are getting reproductive disorders from endocrine disruption. And the reason is, when we look at sex determination in mammals, in humans, we start out with a chromosome, which you learn in school, and then there's some genes on the Y chromosome that determines if you get a testis or an ovary. So this is genetically determined. And then you get different gonads. After that, most of what happens is regulated by hormones. So all the other tissues, all the other organs are influenced by the hormonal milieu, and that drives this sexual differentiation of all the other tissues, including the genitals. So that then makes sense. If you disrupt this endocrine system during this critical life stages, development is not progressing normally and you get disorders. And this is basically what we know. And then when we get to toxicity testing for endocrine disruptors, we also know the mechanisms driving masculinization, feminization, and it's mainly androgens that sort of drives the masculinization programming window, and that is testosterone produced by the testes, secreted into the system, and it acts on the androgen receptor in peripheral tissues, but you also have this very important enzyme called 5-alpha reductase that converts to DHT, dihydrotestosterone, which is more potent androgen receptor ligand, that is really, really critical to making the male phenotype. So this is a mechanism we know. And now I'm going to do a huge jump, because that's also what NAMS people sometimes do. This is then what we know, and we make tests for them in the lab. So I know that sterogenesis is important. We take the cholesterol into the cell, and you have sterogenesis. You make the progesterone, you make the androgens, you make the estrogens. We can have an assay for sterogenesis, and we do, H295. That's in OECD test guideline system. I know that androgen receptor is important, estrogen receptor is important. I made reporter gene assays, and I can test chemicals if they affect this mechanism of action. I have enzyme activity assays and so forth. So I have all these tests that we can use. So if I'm taking then in this roadmap, moving away from animals, I can just take these tests and say, well, if these are affected in the lab, in the cell, then I disrupt androgen signaling, and I get these adverse outcomes. That is nice in theory. So what we normally do in toxicity testing, we expose animals, and then we see if we see these effects coming up. But if you take the animal out of the question, we have to predict it. So the question then is, can you, based on your knowledge from mechanistic knowledge of development, testing using these very simple assays, and predict everything that is in between, disrupting an androgen receptor and what happens in an adverse outcome in a human? I would say in some instances we can, in some instances we can't. And this is the challenge. And from what we know from experience is that the current panel of test methods we have, they are not good enough to really predict it to replace completely. And this is the challenge. And for me, the challenge is biology. You know, endocrinology is very difficult to predict. You say, well, androgen receptor. Well, androgen receptor acts different in all different cells and tissues because it has cofactors, it has epigenetics, all these different things. You can't just say that's what happens. And this is sort of some of the challenges that we're dealing with the test guideline system. ADME, of course, so, you know, metabolism, distribution, and so forth of the chemicals. All toxicologists know this. You don't need to, you can read about it on Google. Then you have this spatiotemporal understanding that I think is very important. So hormones do different things at different life stages. If you put it very broadly, some of you might chop my head off for saying it, but during development, hormones make things, and during adulthood, hormone makes things work. All right, so it builds the reproductive system during development, and it makes it work in adulthood, right? So they have totally different functions. So this have to be incorporated in when you're testing for things. And I'm also a very big proponent of emergent properties. This is an old theory that comes from physics, astrophysics, emergent properties, but I like to bring it into toxicology. And this is like the whole is greater than the sum of its parts. So it's sometimes very difficult to predict what happens when things are complex. You know, some of you have teenage children back home, and when they're home alone, they're, oh, and then they get together with 10 others, and oh, you know, that's just, we act different if we are in groups, and cells do the same, right? So this is the challenges we have. Very briefly, I could have talked about this for half an hour, maybe more. But then some of the opportunities. So I said we have these assays for very basic mechanisms of action. They are very poor sometimes to predict, especially if we're going into risk assessment where exposure and everything is involved. Hazard may be a little bit better. But we have these challenges. But with challenges, there are also opportunities, right? And some of the opportunities I say, or think I see, is we can't rely on these very simple assays. We have to make more complex assays that are as similar to the in vivo environment as possible without being in vivo. And organoids for me is very promising. And organoids can come from an in vivo. You can take an organ out, of course, but you can also make them from stem cells. You can make them from other organs. So in my field, in testis organoids has come a long way, and I know also the ovary organoids have come a long way. A lot of that work comes from fertility preservation, but we can use it in toxicology. And they can now, you know, they make testosterone, they make all the androgens. They're trying to make sperm and so forth in vitro. So this is, I think, where the research should really focus and put a lot of money into these new assays. You know, stem cell derived co-culture systems, make them complex to make them predictable. And of course, even though I was born now in the early 1970s, so according to the Flynn graph, I'm doing quite well. Um, it's all disasters is some positive. I might be an outlier. In any way, this AI and the systems biology, which is a little bit beyond me, but the young people, they program things. I just last week learned how to do transitions in the PowerPoint, so I'm not the one to do this. But I think it has a lot of opportunities with this new AI power and predictive toxicology to bring this system together, because that's really what we need to do. I got 10 seconds to go. And now I was going to talk a little about our Merlin project and how we do this, how the EU is working on implementing NAMs for toxicity testing. I just have the slides. I don't have the time to go through it, but basically we have a way to go, but legally we can move that direction. So that's good to know. So to not hold you from your coffee, you don't need to read. Just a nice figure. I made it myself. And then just remember, reproduction is a life cycle, and we need to look at everything, especially the fetus. And thank you very much.